Hello, everyone, and welcome. I'm happy to share with you that today's webinar is a joint collaboration with Johnson Matthey, and thanks to them, we were able to bring to you today the webinar on multi-component pharmaceutical crystals from solid form selection to controlled crystallization. Our presenter today is Dr. Ruth Lund from Johnson Matthey, and my name is Carmen Guguta, your host from Technobis Crystallization Systems. Before we start, I would like to share with you a bit of information about this webinar. It is being recorded and the recording will be made available to you tomorrow. You will receive an email to the link. Also on our website, as all our webinars will be available anytime to be able to watch the webinar. You are being muted, but if you have a question, please feel free to make use of the chat function of this webinar platform or write to us at info@crystallizationsystems.com. Also, after the webinar, please feel free to still send your questions either to us or either to our presenter, Ruth from Johnson Matthey. The webinar will last about 45 minutes, including questions, I think. For those that they do not know us, Technobis Crystallization Systems, we are a Dutch company located in the north of the Netherlands, a world-leading technology provider for solid-state research, process development, crystallization process development, and formulation. We are in business since about 2005, and we have more than 500 instruments installed worldwide. Our products, in our product portfolio, we have 3 products, the Crystal Breeder, the Crystal 16, and the Crystal Line. It might be that some of you already have a Crystal 16, a Crystal Line, or even both of them in your lab, or a Crystal Breeder. If after this webinar you would like to test one of these instruments, please do not hesitate to contact us either directly via the website or send an email, as I mentioned earlier, at info@crystallizationsystems.com. Further, please remember, Crystal 16 is your instrument for solubility, metastable zone determination, solid state type of screening, any type of solid state screening. The Crystal Line with the camera technology and that works like a microscope, and the Raman, the connection to a Raman spectrometer, helps you to develop or optimize your crystallization or formulation process. The Crystal Breeder, with its gadgets, the vapor diffusion setup or the controlled evaporation setup, the gadget instrument, as I like to call it, will always help you to grow single crystals and to perform crystallization screens. So if you'd like to hear more about this, contact us directly via the website is the most easy way to do this. And now we are getting to our presenter today, Dr. Ruth Lund. She has joined Johnson Matthey almost 2 years ago after she finished her PhD at the University of Bath as part of CMAC, a National Center for Continuous Manufacturing and Crystallization. During her PhD, she focused on crystallization development in batch and continuous platforms. Since she joined Johnson Matthey, Ruth has continued to work on challenging crystallization development projects alongside solid form screening. And I'm pretty sure you are going to hear much more today. Ruth, thank you very much for doing this, for sharing your knowledge and your experience with us and with our attendees today. I'm giving now the floor to you. I'm making you a presenter. And I think now you should be able to share your screen. Perfect. Yes, we see your screen. Perfect. Thank you, Ruth.
Perfect. Thank you very much, Carmen. Thank you, everybody, for taking time out of your day to attend this talk. So as Carmen said, I've been part Johnson Matthey Pharmorphics for just under 2 years. So within Pharmorphics, we've got 18 years of experience within that solid form area. So we take over 120 projects a year investigating this space. We work on things such as solid form screening, so whether that's pharmaceutical, polymorphism screening, cocrystal screening, or salt screening. We also do a huge range of projects based in crystallization development. So these span from very early stage crystallization, just designing those processes, to further optimization, scaling up these, and also troubleshooting current crystallization processes that might be seeing problems during their scale up and manufacture. Within Polymorphics, we also have a huge range of expertise based in formulation and particle technology, and it means that we really are experts in designing processes that not only control our solid form, but also our particle attributes as well. And within Polymorphics and JM, we've actually worked on over 21% of all FDA-approved small molecules in the last 7 years. So we've got a really good wealth of experience and expertise within our team. So today, during this talk, I'm going to be focusing on solid form and, as Carmen said, crystallization development. So for some of you who might be quite new to the area of solid form and subsequent crystallization, I'm hopefully going to introduce this, and then we're going to have some case study work going into more detail in terms of crystallization development as well. So why is solid form so important? So if we think about a simple molecule or an API, so an active pharmaceutical ingredient, we can have different solid forms of this, and those different solid forms can display really significant differences in their physical properties. So we've got a list of these here, and these are just a few examples of the properties that can differ depending on solid form. And as you can imagine, these differences in their physical properties will have a really significant impact on not only the drug performance, so how it's acting when we've got that final drug product for that patient, but also the processability of that drug as well when we're thinking of things like secondary processing, formulation, and subsequent manufacture of that final drug product. So when we think about our API, it's really important to understand what solid forms are out there, how they interact with each other and those properties, so that we're making sure we choose the solid form with the most desirable properties that are applicable to our final product. So in terms of solid form, if we think about a single API molecule, we can have a variety of different forms. So if we just think about the API molecule itself, these can pack together within that solid state in different packing arrangements. So we can access different crystal structures for a single molecule, and that's what we call polymorphism. So we can see that shown here on the left-hand side. So we've got a different packing arrangement of those molecules. We'll have different interactions within that solid, and that's what directly impacts our solid form properties. So we'll see differences in things such as our solubility, our thermal behavior, or compressibility based on the different forms. But we can also start to think about multi-component systems. So as Carmen said, the title of this talk is Multi-Component Pharmaceuticals, from solid form to controlled crystallization. So when we're thinking about multi-component systems, what we can do is we can start to introduce secondary molecules into those crystalline structures. And this really opens up a whole new area of different solid form properties by doing this. So if we think about our API, what we can have is if we have an ionizable center on that API molecule, we can start to introduce counterions where we would have complementary pKa differences, and these could crystallize together with our API in the crystal structure, and we'd see hydrogen transfer happening between those molecules, and we'd form salts. If we don't have an ionizable center in our API, or potentially we're using secondary molecules that don't really have a complementary pKa value, we can still get incorporation of those molecules into our crystal structure. And that's what we call cocrystals. So within this, we're just forming favorable intermolecular interactions between those molecules. We can also start to think about incorporation of solvates. So the most applicable of these is obviously going to be hydrates when we're incorporating water molecules into our crystal structure as well. And something I'm not going to touch on this talk is also the awareness of amorphous solid forms. So this is where we have solids without any long-range order. In some cases, these can be more favourable. They can offer much more enhanced solubility, but there can be some issues with stability. But as I said, we're not going to be focusing on that. We're going to be focusing on these crystalline solid forms. So if we start to think about an API, we can really clearly see how for a single molecule, we can actually access really complex solid form landscapes. So there can be a huge variety of different polymorphs, and there can also be a huge variety of salts and cocrystals and hydrates as well. And all these multi-component systems themselves can also display polymorphism. So we can quickly see how we could access a huge range of solid forms, and that can become quite complex. But the key to when we think about our API development is really understanding that landscape, knowing what solid forms are out there, and understanding their properties. And once we conduct these screens, which due to time, I'm not going to really talk about what we do with screening, but we can conduct screens, understand those solid forms and fully characterize them. And once we have that characterization data, we can look at which solid forms are offering those desirable properties and which ones are going to be the most applicable to subsequent scale up and development. So we'd go back to these properties here, and we'd just make sure that we're accessing the solid forms based on the drug performance and the processability of that as well. So what I'm going to introduce to you now is the kind of case study system that majority of the data we've collected and I'll be talking about for this talk is based on. So within Pharmorphics, we very heavily use pyrimethanol. This is actually an agrochemical, but it's now just 2 APIs. And as a model system, it's really lovely. It has a huge range of different co-crystals and different salt systems as well. And the system we're going to be talking about today is a multi-component system with pyrimethanol and L-tartrate. So this multi-component system actually has two solid forms. So the first one we can have is on the left here, so we have a salt. So within that crystal structure, we have a 1-to-1 ratio of our pyrimethanol to that L-tartrate, and we see hydrogen transfer between those two molecules. This is the thermodynamically stable form, but we can see really clearly from that SEM image that we've got really undesirable fibrous morphology, and this has subsequently really horrible particle handling. So what we can see in the middle is an upturned vial of this, and you can quickly imagine that if we were taking this forward and scaling up, they're really undesirable handling properties. But for this multi-component system, we actually have a second solid form. So this is a cocrystal, and we have a different stoichiometric ratio of those two components. So now we have a 2-to-1 ratio of our pyrimethanol to our L-tartrates. This is metastable, so it's the least stable form, but we can see from those SEM images, we've got a much more desirable particle morphology. So we're accessing nice block-like particles. And this really lends itself to having much more desirable handling properties. And so in terms of this case study, this is the system that we're taking forward, and we've decided on this desired solid form, and we want to subsequently be able to isolate this robustly and scale this up as well. So when we start to think about isolating our material, we need to think about crystallization and crystallization development. So when we're thinking about crystallization, it's really important to be aware of all the different things that you need to be able to control so that you're getting a really nice, scalable, robust process. So we need to be thinking about our solid form. Obviously, we've done a lot of work on solid form screening, and we've decided on our desired solid form. We need a crystallization process that's going to be giving that exclusively, and that's really important. We need to be thinking about things such as our yield. We want to be optimizing and maximizing our yield from that crystallization process, whilst also taking into account things such as our purity. So we want to be purging the impurities from our system, selectively crystallizing our API and leaving those impurities in our mother liquor. Also thinking about things like our solvent entrapment. So API batches can fail because their residual solvent levels are above those ICH solvent limits, and they would subsequently need reprocessing. So to save time and money, a crystallization process that reduces that residual solvent is obviously beneficial. Our crystallization processes can also be really helpful in terms of particle handling. So this will, on some level, be controlled by our particle, our solid form we've decided, but by manipulating our crystallization process, we can exert some control on our particle size and particle size distribution, which is really important. We also need to be thinking about kind of untoward things that might be happening. So an example that I'm going to talk about in more detail later on is oiling out, and that's something that we really need to be aware of early on, and we need to find routes around that to make sure we're not getting it. It can be really detrimental when we're working at large scale. And always in the back of our minds when we're developing crystallization processes, especially very early on during an API's development within that pipeline, we need to be thinking about scalability. So even at the small scale, we need to be thinking about how we can reduce those solvent volumes and those batch times so that it's more scalable as we move forward. So obviously I've just listed a huge kind of area of outcomes and target parameters that we want to be focusing on and making sure that we kind of control and can manipulate our crystallization to give these target parameters. But how do we actually do that? So we really need to understand what's actually going on during that crystallization. And the first thing we need to start measuring is our API solubility and gaining a really good understanding of this. So for those of you who potentially are quite new to this area and aren't so familiar, I'm going to go through the kind of solubility in a little bit more detail. So what we have here is a solubility curve in pink. So this is giving us our information of the maximum amount of our API that we can dissolve in our given solvent system at any temperature. And we can see a really nice temperature dependence of this curve. So as we increase the temperature, we're able to dissolve more API in there. If we're at a point where this laser is, so to the right-hand side of this curve, below it in the white area, our solution is undersaturated. So we have less API dissolved in there than our thermodynamic solubility value. If we were to then take this system and cool it down, we would cross over that solubility curve and we'd cross into this area that's called the metastable zone. So within this area, our solution would stay a nice clear solution, everything would be dissolved, even though our concentration is now actually above that solubility value. So our system is now known as supersaturated. We've got more API dissolved in that than we should based on our solubility. And this is because there is an energy barrier for nucleation, which is the birth of new crystals in our system. And because of this energy barrier, our system stays as a clear solution, and we'd need to keep cooling it down, increasing that supersaturation, increasing the energy within our system so that we can eventually overcome that kinetic barrier to crystallization. So once we cool it down, we cross over this metastable limit and enter the labile zone. And this is the area at which we've overcome that energy barrier and we get spontaneous nucleation. Once we get that nucleation, we can get subsequent crystal growth and our concentration will eventually drop back down to that solubility curve value. So what we really need to understand is when we're starting to design a crystallization process is get a really good understanding of that solubility curve in a range of different solvent systems, but we also need to be very aware of that metastable limit. And that's something that hopefully as we go through this talk and we talk more about that crystallization design and development, that will become very clear, because we need to understand that metastable limit, we need to know where it lies, but the main thing for that is to be very aware that this is kinetically controlled. So although our solubility is thermodynamic and that will remain the same for a given solvent system, our metastable limit can change. So as we're moving through crystallization design, changing anything that can change the kinetics in the system, this can be quite variable and move, and that's something that's really important to remember. So in terms of solubility measurement, for the majority of systems, projects that come in, we use the Technovus Crystal 16. So Carmen touched on this equipment to begin with in the introduction. This is a really useful piece of equipment to be able to generate really nice solubility curves for our systems. So how we do this is we collect what's called clear and cloud point data for our API in a range of our different solvent systems or solvent anti-solvent systems over a variety of different solute concentrations. And we can collate this data to generate a solubility curve. So what we do is we take our systems of known concentrations and we heat them up. And whilst we're heating them up and subjecting them to this temperature profile that we can see here in blue, our material will start to dissolve. And once we reach our solubility value, we'll get full dissolution, and a transmission of a laser through that vial will then reach 100. And at that point is our clear point. That's giving us our solubility value for that particular concentration in that solvent system. And we collate all of that data, and what we can do is build up this really nice solubility curve that we can see on the right-hand side. We subsequently subject those same samples to a cooling profile, so we cool them back down, and if they spontaneously nucleate, we will see that transmission of the laser drop back down as we no longer have a clear solution. And this will give us our cloud points, which again we can collate together, and it will give us information of our metastable zone model. So once we've accessed our solubility in a range of different solvent systems, what we can do is we need to start thinking about which one is the best. It's going to give us the best crystallization process and kind of the start and end point of that crystallization. So what I mean by this is I'll go through a quick example to explain. So what we have on this slide here is some solubility curves. So in green, this is solubility of our API in a neat solvent system, and the blue line is the associated metastable zone width. And what we can see is if we were to take initially a 10 volume process, so that equates to 100 megamole concentration. So we can see from our solubility curve that this dissolves around 40 degrees. So we heat up our system to 40 degrees and 10 volumes of solvent, we should access a nice clear solution. If you would then cool this down and allow crystallization to happen, we'd eventually end up at this lower point here. So around 5 degrees, our solubility is about 40 megamole. So what we should access is 60 megamole. Sorry, it's just stopped sharing. Carmen, is it still sharing? I think I'm having IT issues.
Now we see the slide. Try to go to the next.
I'm not sure what happened there. Apologies, everybody. So if we think about at the end point of this crystallization process, we're now at 40 megamole. So we've accessed 60 megamole from that solution out into the solid. But that's actually only 60% of what we put in there, which when we're thinking about crystallization is a pretty low yield, and we definitely want to be optimizing that to get more material out of solution. So there's a few different ways in which we could do this. One way was we could stay within our neat solvent system, but we could use higher concentrations. So now we're working at lower solvent volumes, we're going to higher concentrations. What this means is that we'd need a higher temperature to dissolve this, but as we cooled it down, that 40 megamole that's remaining in our mother liquor is a lower percentage of what we put in. So in that way, we've increased our yield. But there's a few issues that could happen when we think about this. We could start to be working in kind of too low solute volumes, so we have very thick suspensions at the end, which are very difficult to manage and isolate, or we could be going to temperatures that just really aren't applicable when we're thinking about scaling up and manufacture. So one way around this is by changing the destination of our crystallization process, and we can start to introduce anti-solvents into our system. So anti-solvents are any solvent systems where we see really poor solubility of our API. And by introducing those into our crystallization system, we can reduce the overall solubility of our API and increase and maximize the yields in that way. So what we can see down here in dark blue is the solubility of our API in a one-to-one solvent to anti-solvent system. And we can really clearly see that huge drop in solubility as we move from neat solvent to the one-to-one ratio. So again, if we think about starting with a 10 volume process in our neat solvent around here, and we add our anti-solvent, what we can do is eventually end up all the way down here, cool it down, so we're around 15 degrees, and our solubility is now about 5 megamol. So in a best case scenario, obviously depending on kinetics of the system, we could potentially be accessing a 95% yield, which you can clearly see is much better than the yield we'd be accessing if we just did a cooling crystallization. So to really understand this and be able to determine which solvent systems are best and which anti-solvent systems, we really want to model this solubility data. So to do this, we use Dynochem. We have a crystallization toolbox, which is really useful. And what we can do is we can take all those solubility data that we've accessed from our Crystal 16 experiments, and for a particular solvent, anti-solvent system, we can regress this together as a set. And it allows us to visualize that solubility not only as a function of temperature, but also anti-solvent. And we can model that globally. This is really important. It allows you initially to distinguish between different solvent systems, and the toolbox itself allows you to start to design crystallization processes. So it will give you information on what anti-solvent percentage you need to get down to and what temperatures to maximize your yield. And it's really useful because it determines that impact of dilution on your yield as well. One really important thing that it does, which can be really beneficial when you're trying to decide between different solvent systems or different anti-solvent systems, is it gives you invaluable information about synergistic solubility maxima. So if we look at this example on the left-hand side, what I mean by this is, for this case, with this solvent, anti-solvent system, we see maximum solubility in the neat solvent. And as soon as we start adding the anti-solvent, we can see this drop in solubility happening really nicely. But in some cases, for particular APIs or different solvent anti-solvent mixtures, we actually see, as we start to introduce anti-solvent into our system, an increase in solubility. So kind of the opposite of what we'd be expecting. That solubility increases until it reaches a maximum, and then as we start to charge more anti-solvent, that solubility will decrease. And it's really important to know where that solubility maximum is so that you can choose the best starting point for crystallization and understand what's actually happening during that. So once we've accessed all that solubility data, decided on our solvent system to take forward, what do we need to understand next to be able to exert more control over our crystallization process? So what we really need to be thinking about next is measuring that dissolved API concentration and how that evolves with time, because that gives us key information about the kinetics. It can allow us to design crystallizations for control and also determine things such as our particle size as well. So if we go back to this solubility curve, what we have here, so we've got solubility and metastable zone width, and we've got 3 different pathways. So these kind of 3 different concentration profiles that could be happening during a crystallization. So the first one in blue, number 1, it shows when we have an unseeded crystallization with a rapid cooling rate. So what we can see is we start from an unsaturated solution and we cool it down, and we cross into that metastable zone width. We keep cooling it down until we reach that metastable limit where we get spontaneous nucleation. As soon as we get spontaneous nucleation, we're removing API from that solution phase into the solid phase. And so we see a drop in the solution concentration. But if we're cooling very quickly, we're still generating supersaturation within our system. And if we're cooling quickly, we're generating this much faster than it can be consumed by the crystallization kinetics of that system. And as a result, our concentration profile tends back towards this metastable limit, and we see spontaneous nucleation happening again. This can keep happening throughout our crystallization process, and it means that we actually have no control. So every time we have spontaneous nucleation, we could be nucleating an undesired solid form, we could be trapping impurities, we could be trapping solvent, and we have no control over that. And when we start to think about particle size as well, we've got no control over that either. We're going to have a very wide particle size distribution because we're continually generating nuclei and fines within there. The other extreme of this is number 3, so this green line here, where we have a seeded crystallization with a very slow cooling rate. So again, we start with our unsaturated solution, we cool it down so we get into that metastable zone, and we've got a supersaturated solution. At this point, we then add seed particles. So we add particles, crystalline particles of our desired solid form. And by adding that, we kind of bypass the need for nucleation, and we can allow the supersaturation within our system to be consumed by crystal growth on those seed particles. So we add our seed particles, and our concentration of API in solution will subsequently drop because of that crystal growth. If we then cool down very, very slowly, any supersaturation that we're generating, as we're cooling, is immediately consumed by that crystal growth. And so our concentration profile will stay very, very close to that solubility curve all the way down to the end point of our crystallization. In an ideal world, what we really want is to optimize this. So we want to understand those crystallization kinetics so that we can match that cooling rate to match those kinetics. So we're following pathway 2. So we can cool at a much faster rate. We can keep a fairly constant supersaturation throughout our crystallization and allow it to tail off at the end. And this means that we can reduce batch times. But to understand what pathway we're taking and to be able to exert that control, we need to understand the kinetics, and we need to be monitoring the evolution of our concentration in situ. So to be able to do that, we need to start to use process analytical technology. So for our crystallization processes, we tend to use the Blaze Metrics probe very heavily. So this is a really powerful tool. It allows you to get multiple in situ measurements from a single probe. So we can get chord length information, those distributions into statistics. We can get information about the dynamic turbidity. We can get high resolution optical microscopy, which is really invaluable when we start to think about we can visually see what's actually happening within there. And also something I'll touch on at the very end of this talk is particle focused Raman spectroscopy, which allows you to understand and see what solid form you've got going in there as well. In terms of our solution concentration, we'll be using the Mettler-Toledo React IR. So this is an in situ IR probe, and it allows you to monitor the dissolved concentration in situ in real time. And it's optimized for use in the mid-IR fingerprint region, which is really applicable when we're thinking about our API systems. We generate all this data, and to further understand it, we use the Dynachrome Crystallization Toolbox, and this allows us to model the kinetics of the system and understand them and design really robust growth-dominated processes that are also very scalable. And by utilizing this process analytical technology in combination with that Dynachrome Toolbox, it allows you to speed up that development as well by reducing experimental burden. So we start to think about the React IR and how we go from measuring that the IR spectra in solution to giving us the nice dissolved concentration. We need to generate a model. So how we do this is probably about, on most cases, a few days worth of work. We generate a nice calibration and use this as a model. So we get in situ IR spectra using that probe for a range of different API concentrations over a range of different temperatures. So on the left, we can see the location of data points that we've collected to generate this model. We then start to look at both the concentration dependence on that spectra, but also our temperature dependence as well. So what we can see here is our concentration dependence. So for a particular temperature, 40 degrees, we've plotted the spectra for that range of API concentrations. And we can start to see those peaks that are unique to the API system, and we can start to understand how that peak intensity changes as we increase the concentration. It's really important then to also look at our temperature dependence. So what we can see at the bottom here is for a particular API concentration, our spectra collected at different temperature. And we can start to see that as we change that temperature, we see differences in the peak intensity and the peak broadening. And it's really important to use that and put that into our model and generate a multivariate model for our data to get a really robust model, so that when we're collecting that spectra in situ, it can tell us a very accurate value of our API concentration. So once we've designed that model and we've got a really robust one in process, we can then carry out crystallizations using that, and we can follow our concentration evolution. So what we can see here on the left is just for a simple cooling crystallization. So we can see our solubility curve and our metastable zone model, and this light blue is our concentration. So what we've done, we've cooled down, we've generated supersaturation, and we've added seed to our system, and we've held this isothermally, and we can see that during that isothermal hold, that drop in concentration as we're getting growth and absorption of that supersaturation on our seed crystals. We then start to cool down. As we're cooling, we're generating more supersaturation within our system. But we can see from that concentration profile that as we're generating that supersaturation, our crystallization is continuing and we're consuming that, and we're following the solubility curve really, really nicely down to our endpoint. And we have another isothermal hold and our concentration drops to that solubility value. So what we can see initially from this is that we're in no way encroaching on this metastable zone. So we should be having a nice growth-dominated process. We shouldn't be having any kind of untoward nucleation events happening. What we'd ideally want to do is make sure that we're using the most optimum cooling rate for this. And to do this, to understand those growth kinetics, we can do some really simple isothermal desupersaturation experiments. So for this, we would take solutions of a known saturation, both at high and low temperature, and we would add seed particles in there. So we'd add a known amount of seed of known seed size, and we'd watch how that concentration drops over time. So what we can see on the bottom here, we've added our seed and we can see that concentration dropping down, and it starts to plateau about 15 minutes. So we've got full desaturation within 15 minutes. By doing this at both high and low temperatures and then inputting this information into Dynachem, we can get some really nice model fit, and we can model those global growth kinetics. And once we understand that, we can start looking at how we can optimize those cooling rates. And we can also then use this to model our particle size distribution as well, which is something I don't really have time to go into today. Once we've got a full understanding now of our solubility and our concentration and which pathway we're taking throughout our crystallization, we really need to understand if anything else is going on there. And the best way to do this is just by imaging. So if we look back and think about those process analytical tools, we're using the Blaze probe, and that gives us optical microscopy, and we can see exactly what's happening on, which is really key. So the example I'm going to talk about today, which is something that we see quite regularly on the crystallization development processes we do here, is oiling out. So for those of you who might not be aware of what oiling out is, I'll give you a quick example and then we can discuss why this can be detrimental to our crystallization process. So when we see oiling out, what we do is we go from this solution of dissolved API to a suspension of droplets within our system. So these oiling out droplets are rich in API, and it results in having an API lean continuous phase. But within these droplets, because we've got a much higher concentration of our API, what can actually happen is spontaneous nucleation. This is something obviously we've discussed and we want to avoid. So we want to avoid the spontaneous nucleation happening, and because we're losing control over our system as soon as this happens. At the small scale, this can be really difficult to see just visually by eye in a vessel, and potentially if we don't spot this early on, we can scale up and we can have these issues happening, and it can cause really huge issues within our crystallization process. But by having process analytical technologies, we can visually see this. So what we have here is images taken from a process where we have oiling out with the Blaze probe. So we can see the suspension of those oil droplets really, really clearly by using that probe. And over time, we start to see nucleation and then subsequent crystallization happening in there. So in terms of oiling out, it doesn't mean that process is kind of over with. There's many things that if we see this happening early on, we can change within a process and understand. So firstly, we need to understand why this might be happening. So there's a variety of different reasons we might see oiling out. So it might be that we have quite low melting point API solids, or our APIs are quite solubilic, and it can also be induced by impurities. So there's a few things we can do to avoid this. So depending on what stage we are in the drug's development, we can potentially purify. So it might be something that naturally happens as the upstream processes change and that impurity profiles change, or it could be something we could look at having an initial purification step before our final recrystallization. We could also change our solvent system. So we could look at solvent systems where we're not as solubilic. And one thing that can really be indicative of oiling out in a particular solvent system is when we measure those solubility curves, if they're very exponential and it looks as if they're reaching an asymptote, that might give you an indication of oiling out. So even early on, we can start to eliminate particular solvent systems. If we're kind of tied to our purity profile or our solvent systems, we can also look at seeding as well. So we can start to change where we're seeding, how much seed we're adding to try and circumvent that oiling out as well. Finally, what we need to think about is any changes in solid form that might be happening as well. And we really need to understand that so that if this is happening and it's a problem that we're seeing, we can look at ways to circumvent this. So going back to our model system, this L-tartrate system of pyrimethanol, we have a monosalt and a hemicocrystal. And hopefully you can remember that we have two very different morphologies for these. So this system is really nice because of that two different morphologies. It's something that we can visually assess which solid form we're having. So again, we've got some nice Blaze images from a cooling crystallization process where we started with that hemicocrystal, but very quickly we start to see the formation of this fibrous morphology, and we can see the conversion from those block-like crystals to that fibrous morphology, really showing us that we've got that solid form conversion happening within our vessel. But for a lot of systems, we might not have solid forms that have a visual difference in their morphology. So we wouldn't be able to visually monitor this. And we'd have to look at a different way to understand what solid form we have. And that's where Raman spectroscopy really comes into its own. So what we can see here on the top is our Raman spectra. So we can, for our system, we have a peak shift, which is highlighted here in pink, moving from our hemicocrystal to our monosalt. We can see that these two peaks shift. And we can monitor that throughout our crystallization to understand what solid form we're getting and when we start to see this conversion. So down here, we can see the spectra collected throughout that cooling process, and we can see really clearly that peak shift as we move forward. We can then start to collate this data and look into it in a little bit more detail. We can correlate it for our stability and normalize that spectrum. We can start to see that peak shift really, really clearly in this 3D contour plot. And this gives us really invaluable information because it's telling us exactly when we're starting to see that solid form conversion. And if we understand that, we can then start to put processes into place to avoid this. So potentially we could look at different cooling rates. If it was an anti-solvent system, changing our anti-solvents, changing our seeding regime, also looking at different solvent systems. So the main takeaways from this, as I think I'm really highlighting, is that it's really key to understand your solid form landscape when you are developing an API to make sure you're getting the most desirable and applicable solid form. And once you've understood this and designed it, you want to be able to design a robust, scalable crystallization process. And to be able to do this, you really need to get a full understanding of what's happening. And this really is only, we're only able to do this if we're using PAT in combination with modeling. What we always say is it's never too early to start crystallization development. So by identifying all of these issues that we've discussed today, especially things such as oiling out, solid form conversion, and overcoming them at the very small scale, allows us to then scale up and maintain that process and product integrity as we move forward. So that's everything I wanted to talk to you today, just kind of highlighting acknowledgements to those people who are really instrumental in kind of getting all this data and putting it together so that I could present it to you today, and just to highlight a white paper that was written by Joe Harris, which gives a kind of deeper insight into the crystallization process as well. So thank you very much for your attention, and I'm very happy to take any questions you might have.
Thank you very much, Ruth. Quite a nice presentation with a lot of interesting data and case studies. I also liked those images of the transforming from one form to another one that you have taken. It's quite a nice case. I have quite a few questions I see here. I will try to read some of them to you. One of the questions is related to, well, actually we have a few questions related to the Dynochem use, and they are all related. If you can elaborate a bit more on, share a bit more details on the use of the Dynochem.
Yeah, so with the Dynochem, there's a huge range of different toolboxes you can use. So for this one, it's the Crystallization Toolbox. So once you've generated, you've got all your clear and cloud points from the Crystal 16 data for a particular solvent or anti-solvent system. What you can do is you can input that into the Crystallization Toolbox, and it will model your data for you. So it can give you a really nice fit of your solubility, and it gives you kind of a range of different modified Van Hoff equations, and it can tell you which one gives you the best fit as well. And once you've done that, you can then use the Crystallization Toolbox to look kind of at your starting concentrations, what volumes you want to be working in, how much anti-solvent you need to start adding to meet your desired yield, and also your cooling and final temperatures. And then you can also then subsequently use this. So when I was discussing the growth kinetics, you can put in your information from those desupersaturation experiments, and it will model that, and it can tell you about your growth kinetics. And then subsequently, because you've known how much seed you're putting in and the size of that seed, it can then give you information about, it can give you models about your particle size distribution, and you can also refine those models as well. So you can do a lot of offline measurements of your particle size and put that back into Dynochem to refine your model to take into account anything else that might be happening, such as attrition and agglomeration. And it's a really useful tool to help you design those processes robustly.
Yes, thank you. Another question related to crystal morphology. The question is, which crystal morphology is best for good flow properties of the solid? I think it's more than the morphology.
Yeah, so ideally you want quite like block-like morphology. The issue is when you have a very fibrous material, acyclic material, that kind of all clumps together and it doesn't really flow very well. So having nice block-like morphology is much more desirable. Obviously, in some cases, that's quite difficult to get just based on your solid form. It might kind of have the desired morphology of being acicular, but there's things you can put into place. So you can look at things like temperature cycling to kind of reduce that aspect ratio, or things like milling as well.
Yes. Another question related to poor soluble systems, compounds, I guess. What are some of the specific characteristics of poor soluble compounds is the question, but I think what our attendee would like to know is related to what would you do actually with a poor soluble compound? What would you suggest? What would be your strategy related to a poor soluble compound?
So if your API has a really low solubility, that's when you can start to look at multi-component systems. So by looking at co-crystals or salts, you can enhance that solubility to a much larger amount than you can in terms of different polymorphs. So that's definitely something you should look into.
Yes, definitely. I completely agree with you. I have seen many cases where, the usual, let's say, workflow would be to look into salts. If you can make a salt with your API, of course, and followed by a co-crystallization. And I think in the last case scenario, in your decision tree, let's say, would be to even go for a more solid dispersions. But of course, it depends on much more things. Okay. Another question: how is the Van Hoff equation modified to be fitted globally? I think it is related to the very first few slides when you were also talking about I think the dynochem.
So within the toolbox, say you put in your data and effectively it fits it. So there's a range of different equations it can use, and it will fit those your data, and it will tell you the best one in terms of kind of like the error and the R value.
I think we have a few more related to dynochem, but I think you have also already answered them. Ruth, I didn't actually, we didn't touch this point. You are going to share the slides with our attendees. I'm not very sure. We need to check this if you are allowed by Johnson Matthey. I don't know if you have an answer yet. If we can promise anything to our attendees or not yet.
I don't think yeah, so a decision hasn't been made our end, but I'll definitely be chasing that up as soon as we finish this webinar.
Okay, perfect. So you'll hear from us definitely in the coming 1-2 days with related to the slides. So there you will be able also to actually see and have access to a bit more of the information shared by Ruth today. Oh, I see now quite a lot more questions are coming in. Maybe I take a few more, Ruth. Do you have any advice? Oh, another one coming, and just one second. Do you have any advice for the selection of a potential conformer?
So it depends on your molecular structure of your API. So in terms of when you're thinking about your molecule, you'd want to look at your pKa. So in terms of whether you're going to be getting tending more between a salt and a coformer, it's that pKa difference. So if you're looking at molecules that have quite similar pKas, you're much more likely to be accessing a co-crystal. And there's loads of information out there in terms of kind of like crystal engineering about looking at what hydrogen bonding facilities you have in your API molecule and choosing coformers that kind of have applicable hydrogen bonding, so things like carboxylic dimers. And it's also really important to look at those which have already been used, so co-crystals that are out there on the market in terms of a filing perspective as well, making sure that you're using molecules that are applicable for regulations as well.
Yes, indeed, there's quite a lot of information out there on the co-crystallization and how to choose a co-former. We also have on our website one webinar performed quite some time ago by Professor Jopter Horst on co-crystallization and how to choose the co-formers. Sorry, it was quite a nice method, also published in an article in 2008 by the same Professor Jopter Horst that was not so long time ago, until not so long time ago at CMAC. Indeed, we have another question again related to the co-crystal, but in co-crystals in general. How would you select your co-crystals candidates? I think it's also related to the objective. What will you look for, right, Ruth?
Yeah, so once you screen a whole range of different co-formers, and then hopefully you'd get some hits from that, and then you would look at those properties. So which ones are kind of giving you the better solubility or the better thermal behavior, and then you kind of look at which ones are the most desirable in terms of your end product.
Yeah. Another question. First, thank you for this comprehensive overview from Luc. And in case of these two forms existing, do you ever establish a phase diagram?
It's not something that we tend to do in much detail, but it's definitely something that there's a lot of information out there of how you can establish those and how they relate to one another. But it's not something we overly focus on. But the mono will always be more stable than a hemi. So in terms of a 1-to-1 ratio as opposed to like a 2-to-1.
One more question related to oiling. Can you discuss the relationship between the oiling potential with the steepness of the curve?
Yeah, so that's a really good question. So it's in terms of the kind of the API, as opposed to being dissolved, it becomes more like it's melted in there. And that's why you then kind of see that steepness. It's not actually giving you a true reflection of your API dissolving, because your API has now become kind of a liquid phase and you're getting that.
Okay, perfect. One last question. I'm always saying one last question. A lot of questions are still coming up, Ruth. You see, it was quite an interesting topic, and all the information that you have shared. People have quite a lot of questions. Is there a way to design a circular, so needle type life curve like crystals to improve the aspect ratio? To design the perfect needles.
Yeah, so you can look at things like introduction of wet milling within your cycle. So you could cool down and then you can cycle your material through. And we've actually had a really recent project which myself and Joe worked on with really good success of that, of doing in situ milling and with some temperature cycling as well, and reducing that aspect ratio down really significantly, and that, as a result, the pore bot density was much higher than that initial material.
Okay. I think we are almost ready, Ruth. Once again, thank you very much for joining us today, for making this webinar available. Thank you very much, all your colleagues at Johnson Matthey that made this available, and I'm hoping this is just the beginning of a longer-term type of collaboration between us and Johnson Matthey. Definitely we see it was quite a lot of interest. Thank you once again, Ruth. Thank you all attendees that you are here with us today, and for all these nice questions and all the good words and wishes to Ruth. We will be back with the next webinar on the 10th of June, on Thursday, 10th of June, a webinar brought to you in collaboration with AstraZeneca and Dr. Sophie Jambou from AstraZeneca. I hope to see you also at our next webinar. Have a good day, good morning, and good evening to all of you. Thank you. Bye.